Dear student, today we will talk about photosynthesis. Now, photosynthesis is a biochemical process in which plants and other organisms, such as certain bacteria, protists, algae, make use of sunlight to synthesize glucose, which is known as food, by using carbon dioxide and water. During the process, the sunlight is converted into chemical energy, that is ATP, and oxygen is released as byproduct. So in a very simple way to understand, the sunlight energy is converted into chemical energy, that is ATP, and this is utilized to convert the carbon dioxide into glucose, and during the process the water is also used up to make the oxygen, which is released from the leaf. Now, to be very precise, the photosynthesis is divided into two phases. One is light dependent phase called light reaction, and another one is light independent phase called dark reaction. So first phase, the light is trapped by chlorophyll molecule, and this light energy produces two different things. First is the chemical energy, that is ATP. Second is NADP, which is known as reducing power. And NADP actually is the coenzyme which is required for oxidation reduction reactions. So we will see how they are useful in the photosynthesis. Then oxygen is released by photolysis of water. So these are the things which are happening in the light reaction. If you see the dark reaction, the ATP and NADP which is produced during the light reaction is utilized in the dark reaction to produce glucose from carbon dioxide. This is also known as fixation of carbon. So we will see how these things are taking place, and before that you need to understand the structure of leaf. So this is a leaf, and if you study the longitudinal section, you will find the upper and lower epidermis, and in between that certain mesophyll cells are there, and the upper mesophyll cells are called palisade cells, which consist of large amount of chloroplast, and chloroplast actually consist of chlorophyll. So if you look at the structure of chloroplast, then you will find that it is a double membrane structure, and it consists of a matrix which is known as stroma. Okay, and it consists of the uh, grana, which is made up of thylakoids. So you can see the grana are connected by stroma lamella, which are known as fruit. Okay, now if you see the thylakoid membrane, this is a site for light reaction, and if you see the stroma, this is the site for dark reaction. So the thylakoid membrane, what exactly, when it is forming a site for light reaction, that means it consists of certain other things. So it basically consists of the various pigments which are utilized during the light reaction. Like we have chlorophyll A, B, C, D, carotenoid, phycobilins, phycoerythrin, phycocyanin, xanthophyll, etc. Okay. Now chlorophyll A is the main pigment, and other chlorophylls are also there, and rest of them are accessory pigments. They all will absorb the light, but finally they transfer their light to chlorophyll A. So they are arranged in a very organized way. Now if you look at the structure, um, in the center there is core molecule, which is the main pigment, consists of chlorophyll, and on the side the antenna molecule are arranged, which are accessory pigment. So the core and antenna all absorb the sunlight, but the antenna molecules, they transfer their energy to the main you can say pigment, and this is known as reaction center. So these pigments all together known as reaction center or photocenter or quantosomes or light harvesting system or photosystem. Okay, so basically it forms the place where light energy is trapped to start the light reaction. Now we will talk about the photosystems. So photosystems are nothing, all these pigments that you see, they are basically all proteins. Okay, so photosystem consists of complex organization of protein, There are main two photosystems. One is photosystem 1 and photosystem 2, which are formed in the green plant, and they absorb the light energy at different wavelength. Okay, like photosystem 1, which absorb mainly near the 700 nanometer wavelength, whereas photosystem 2 at p 680 nanometer wavelength. So they are known as p700 and p680 respectively. Photosystem 1 is located at the outer surface of thylakoid membrane, also the stroma lamella, and contain chlorophyll A, B carotenoids. Okay. Whereas if you look at the photosystem 2, it is located to at the inner side of thylakoid membrane and consists chlorophyll A, B, phycobilin, xanthophyllin, etc. Okay. Now we will see what is there in the light reaction. So during the light reaction, the chlorophyll, it absorbs the light. So which part? The thylakoid membrane, because it consists of photosystems. So chlorophyll actually absorbs red and blue part of light. The green light is not absorbed by chlorophyll. It absorbs and first it reaches to photocenter 2. So the light energy is absorbed by different molecule, and finally they transfer it to the center molecule of photosystem 2. Okay, now once this light energy is gained by photosystem 2, the electrons are excited from the photosystem 2, and these electron will now move through electron transport chain and finally reaches to photosystem 1. Photosystem 1 further passes this electron to finally to NADP, and NADP is converted into NADPH. Okay, now during this transfer, during the electron transport chain, what happens? The various substrate, they releases hydrogen ion in the lumen of thylakoid, and this establishes proton gradient that causes ATP production. Now we will see how this happens. If you look at this as one thylakoid, this is the lumen, this is its membrane. Both the photosystems are present here, and this is how it receives the sunlight. So first we'll just talk about, and outside it is the stroma or matrix. First we will talk about the photosystem 2. So the photons are there, which are the light particles. They are absorbed by P680. Okay, so in P680, whatever accessory molecules are there, they absorb the photons and they transfer their energy to P680. So P680 absorbs the light, you can say, and then it excites the electron. So after exciting the electron reaches to pheophytin. This is the primary acceptor. Okay, now after this the electron goes to plastoquinin. After this, now whenever the substrate they accept the electron, we say this is reduction, so they get reduced, and the one from where the electron are lost is oxidized. Okay, so from the plastoquinin, it goes to cytochrome complex, which consists of B6 to cytochrome F. Okay, and then it goes to plastocyanin, and the plastocyanin transfers the electron to photosystem 1. So try to understand, from P680 the electron now reaches to photosystem 1. Now there are two ways the photosystem 1 channelizes this electron. First is the photosystem uh, the photosystem 1 transfer this electron to FES, that is iron sulfur complex proteins, and then to ferroxidin, and then ferroxidin transfers the electrons to NADP. The NADP is converted into NADPH by the enzyme NADPH reductase. Okay, so now what happened? that energy, the electron which is excited from P680 is finally accepted by NADP here. So final acceptor is NADP. Okay. Now if you look at the second mode, okay, because P700 can directly take the energy from the sun also and excite the electron. So in that case, when it takes the direct energy, the electron goes to FES and then to ferroxidin, and from the ferroxidin it follows a different path and come back to P700. Okay. Now over here you see P680, the electron is lost. Now the electron, whatever is lost, will not come back to P680, but this electron is compensated by photolysis of water. Now again the sun energy is used for lysing the water molecule, which is called photolysis of water, and it produces 4 protons, 4 electron and oxygen, which is oxygen is liberated, and the electrons actually goes to P680, and this compensate the lost electron from P680. Okay, so this is a cycle of events. Okay, now absorption of 4 photons by P680 generate one molecule of oxygen. Okay, now you can see that during this transfer of electron, the protons are released into the lumen. Even during the photolysis of water, whatever protons are produced, they are also released in the lumen. Okay, and once they get accumulated, they generate proton motive force, because the concentration of hydrogen increases in the lumen as compared to the stroma. Okay, and this developed the proton gradient force, and this force helped to derive the ATP synthetase to do the phosphorylation. So we will see next how this phosphorylation occurs. So in the electron transport chain, you can see the electron moves unidirectionally from donor to acceptor through electron transport chain, causing redox reactions, because simultaneously there is oxidation and reduction going on when the electrons are accepted and donated to the next level. So donor are at high energy molecule, high energy level, you can say acceptor are at low energy. Okay, now we will see the photophosphorylation. Now addition of phosphate to ADP to form ATP is called photophosphorylation. The reaction is catalyzed by the enzyme ATP synthetase. When the photophosphorylation occur in presence of light, during light reaction of photosynthesis, it is called photophosphorylation. Now ATP synthase is powered by transmembrane electrochemical potential gradient. Why we call electrochemical potential? Because it depend on the concentration and charges. Okay, so concentration of H plus ion increases in the lumen, so the positive charge increases in the lumen as compared to the stroma. So that's why we say it is the transmembrane electrochemical potential gradient, which is known as proton gradient, and it is due to the difference in the concentration of protons and charges. The gradient is developed across the thylakoid membrane. So this is called transmembrane electrochemical potential. Now this potential set up the electron transport set up due to the electron transport chain, and the electron move from high to low level substrate and causes reduction and oxidation simultaneously. This result in a release of protons from substrate into the lumen of thylakoid, and the accumulation of protons in lumen generate proton motive force that derive ATP synthase to catalyze the conversion of ATP to ADP. So here you can say ATP synthase, it pumped back the protons from the lumen across the membrane to the matrix. Okay, so during this process the ADP is converted to ATP, and the formation of ATP is called phosphorylation since it forms during the photosynthesis. In the light reaction, we say it is photophosphorylation. Here you can see there are two cyclic and non-cyclic. The one which is shown in the right red is the cyclic, non-cyclic, and the one which is there in the green is the cyclic. So if you see the events, the sunlight, it basically falls on P680, which excite the electron, and the electron passes through ETC and reaches the P700, and P700 again through the series of electron transport chain substrate, it reaches to NADP, and NADP is converted to NADH. So finally this electron causes the reduction of NADP to NADH, and it also causes the photolysis of water. But the light is also received by P700, and P700 the electron is excited, it reaches to peroxidine, which passes its electron to plastocumene to cytochrome B6, and then to cytochrome F. Okay, now during these events the hydrogen ion, or you can say proton, they moves in the lumen. Okay, and then ATP synthesis occurs. So this is called non-cyclic phosphorylation. Whereas during this cyclic event where the electron come back to P700, again the because of the same hydrogen ion movement in the lumen, the ATP synthesis derives the synthesis of ATP from ADP. Okay, so two different manner the ATP synthesized, cyclic and non-cyclic, and respectively they are known as cyclic and non-cyclic photophosphorylation. If you see the difference, the non-cyclic and the cyclic photophosphorylation, non-cyclic photophosphorylation involve both the photosystem P700 and P680, whereas cyclic it only involves P700. The non-cyclic involve photolysis of water. There is no photolysis of water in cyclic. The non-cyclic, the electron released from P680 will finally reaches P700 and will not come back to P680. These electrons are compensated by photolysis of water, whereas over here the electron lost from the P700 come back to P700. There is oxygen evolution due to photolysis of water. There is no oxygen in P700 because there is no photolysis of water, whereas in P whereas in a non-cyclic photophosphorylation, the final acceptor is NADP. So NADPH is formed and there is no NADPH production. Okay, so if you look at the net reaction, net light reaction, then two molecules of water are used up to convert into one molecule of oxygen, 2 NADP and 3 ADP plus Pi are used to generate 2 NADPH and 3 ATP. So this is all about the light reaction of photosynthesis.